Our next speaker is Mike LeBlanc. He is the director of the SWOG Statistical Center.
Thank you very much. What I'm going to talk about today is the critical importance of complete follow-up data on our SWOG clinical trials to make sure we have the definitive and conclusive results after the end of everyone's hard work. I presented yesterday at the Clinical Trials Training Workshop, and I'm reminded, and it made me think of actually that as well as looking recently in newspapers, and people talk about data science and big data, and we've all read about that. But we deal with data all the time, and our data, it's a little different than in the public press, right? You hear things about, oh, you've got the fancy algorithm and it's going to come up with answers. We merge all the data. Our data collection is on designed experiments that look at patient care and patient outcomes. And it really is not just an algorithm. It's a team effort right from the very beginning. It's the clinical input, it's the clinical trial design, it's the pharmacy as we saw before, it's the statisticians putting design together in a way that makes it as feasible as possible to collect the data and have it. It's the protocol coordinators to make sure that protocol is actually accurate and has it all steps. It's the data coordinators making sure the data are coming in correctly. And probably the second most important part of the piece are the professionals at the institutions making sure that that data coming in best represents what the patient has consented and given their opportunity to participate in clinical trials. So I'm going to try to capture just one specific topic area, which is the impact of incomplete follow-up. I'm a statistician, so I'll show some statistical pictures and go from there. Oops. Here it is. So there we go. So outline. I'm going to start with a positive result, and I'm going to talk about SWOG 0007, a myeloma study that was actually presented in the Oishi Symposium. So this is like a check back in to say, for all the hard work that went out in the institutions with respect to doing this study, how does it come into the story of good follow-up data? I'm going to remind everybody that statistical algorithms and protocols, it's critical to understand that when statisticians design studies for these conclusive results, they're making the assumption that there is minimal loss to follow-up and minimal differences there. I'm going to talk about an important case and think about what that means. It's just a couple, sort of a hypothetical case to see what that means in terms of loss to follow-up. Contrast that and thinking many of our studies in SWOG, both on the end-course side and the treatment side, really they're randomized comparisons now. And so we think when we do those randomized comparisons that we're having honest estimates between those, unbiased estimates between the arms, and loss to follow-up or inappropriate consent withdrawals, as you'll hear later, can interfere with that definitive conclusion. I'll show some real and hypothetical scenarios because, as I said, I'm a statistician, so we'll present some data. And the conclusions will be, of course, I'll preclude the conclusions, we need good quality data. So here's a picture. So this is SWOG myeloma study S777. It presented, as I said, in the Oishi Symposium about a year and a half back. It was a randomized study. Looking at a 3-drug combination versus a 2-drug combination, and presented by the lead study statistician. So I'll delve into the statistics here. But I want to make a point by showing these pictures. On the far side, you'll see 2 survival curves, a blue curve and a red curve, and the blue curve corresponds to the 3-drug combination. And if you look up from any point, that gives you the probability of surviving past that point in time. So say at 24 months, about 70% of the people are surviving, and this is progression-free, so haven't progressed yet, and about 55% in control arm. So that's a big difference. And if you look at the bottom, you see something called HR, or hazard ratio. That means there's about a 30% reduction in the rate at which these progressive events are happening on the arm. And the p-value, 0.0018, very small p-value. And the reason why I'm going through each one of these numbers is the only reason you can interpret those numbers as being meaningful is because there's good quality data. If you look at this curve, and I'm only spending time here because I'm going to show a couple curves, and you'll see, if you look closely, you see these little tick marks at the bottom. Those mean those are individuals that were lost to follow-up. You notice how they all happen at the end of the curve. That's by study design. That's how the study was designed in terms of when it was designed. You see very few tick marks in the first 4 years of that study. That's because the study has very little early loss to follow-up in terms of these patients. So those really good quality data led to a publication with results that people trusted and really believed that this 3-drug combination actually worked. And just recently, about a month ago, with, again, as I mentioned, subsequent to much work at the institutions collecting additional data to support a regulatory approval, it was approved in Europe, and so now many patients can get access to this combination that wouldn't have been able to otherwise, entirely hinging on good quality data. The sidebar here is the fact that because the data coming in from the institutions was so high quality, the first time around, before we actually went back a second, Antje Huring and Rachel Sexton, the statisticians at the SWOG Statistical Center, looked at the data 2 times, and actually the progression-free survival curves. I'm not going to say that not all that work wasn't useless, but the PFS curves stayed the same. That meant that the scientific results of the first paper held up and provided greater evidence to really show it. So it was all the effort was worthwhile, but it all hinged on good quality initial reporting. And I just want to go through the design here. I'll read it very quickly. I took it from the paper. The sample size was based on the assumption of an eligible patient accrual rate of 110 patients per year, 440 eligible patients, a median progression-free survival of 3 years, control group exponentially distributed progression-free survival, roughly underlying 2.5 years of additional follow-up. The study was designed to detect a hazard ratio of 1.5, approximately 87% power, and overall alpha 0.05. There's no mention of asymmetric dropout or follow-up in this. The statistical design required good quality data, and we had it in this case. So what are critical elements in evaluating therapeutic interventions? Well, they're biologic activity, and all these refer to data points that you're all seeing. Biologic activity, safety and toxicity events, which are obviously, we heard a lot about, are critical at the institutional level to collect that. Clinical response, patient-reported outcomes, disease recurrence, progression, survival, and other long-term adverse event data as well. So those are all elements that require good data coming in to make decisions. And so as I presented yesterday, there are 2 concepts dear to a statistician. It's not sample size and p-values, it's actually bias and variance, because they're the things that stand in the way. Everybody thinks, oh, in the modern world, maybe a tweet stands in the way of the truth. But for a statistician's view, variability and bias stand in the way of the truth, and we have to control those biases and variances. So 4 pictures here as you go from left to right. If a procedure is accurate and precise, that is no bias and no variance, you hit the bullseye every time. If it's accurate but there's variance, if you throw enough arrows and you average all the arrows, you're kind of getting at the bullseye, but they're all over the place. Accurate and imprecise, there's a wind blowing, but you're really good with that bow and arrow, but they're always going to the side. Or accurate and imprecise, you're missing the target, and it's so those are the variability and bias concepts that come up that exist in all of our clinical trials, and our goal is to minimize that. Probably the biggest component is bias, because the interesting thing about bias is it's taking you away from the truth, and in many situations you don't know even what direction it's taking you away from the truth. So the definition of bias is the tendency of a statistical result to differ on average by, you're all nominated as statisticians, you'll all be honorary statisticians after this talk, the tendency of statistical result to differ on average from this true state of affairs due to flaws in design or conduct of a study. That's what it is. And my point, to which I'll get back to, is loss to follow-up can introduce bias because the consent withdrawal or the loss of follow-up, we don't know, might be related to the very thing you're trying to measure. So as a statistician, we can't deal with that. we can deal with missing, in a strange way, we can deal sometimes with missingness if it's totally at random. That increases the sample size, it causes us problems. We kind of have an idea how to, but things that are missing because we don't know why they're missing, that's a problem. And that's what causes studies not to be reviewed highly and not be the results to be used. As I said, worse yet, we don't know the nature of the bias or the direction of it, and it can lead to incorrect results. So I'll just show a quick example here. An illustration of impact to loss to follow-up, and this is a particular patient. Take it patient A, let's call this patient. Registration date, March the 1st. There's an assessment date on May the 1st, and another assessment date on July the 1st, and there's a progression date, unfortunately, on October the 1st. This patient, for sake of arguments, on treatment 1. So let's assume we have a second patient, patient B. It's registered a day later, assessment date, beginning of May, another assessment date in the beginning of July. And then that, as you can see, I colored the arrows here, so that also corresponds to a progression date, July the 1st as well, because those are the dark arrows there. So if you look at the result of this data analysis on 2 patients, it's not a real clinical trial, we only have 1 patient in each arm here. Result of this clinical trial would be progression date on October the 1st for patient A and July the 1st for patient B, just to get what's going on there. However, let's go back to patient A, and we collected all the assessment dates as planned, so we see a progression date on October 1st. Let's consider patient B now, and now let's suppose that patient is lost for some reason on April 1st. So we didn't see those times later on. There's a history where those gray lines are. There's a history of what happened later on, but we never saw it because that patient was lost. The result is we still have the progression date because we had complete data for patient A on October the 1st, but there's no progression listed for patient B. So we've changed the whole interpretation of what those 2 patients told us about that study just because of that loss to follow-up. So this is something we take seriously at SWOG, or everyone takes seriously. And so one of the things we've started doing recently, we've always monitored, but we've just put it more in front of us, is we've created different reporting mechanisms that look at this important issue. So this is a table. You don't see it in our report of studies because typically we see part of this table in report of studies, but the Data and Safety Monitoring Committee and the statisticians see it by arm. We collapse across arms so we don't have a chance of unmasking treatment effects for the public view of it. But if you look in the bottom line here, this is a sort of hypothetical study. These aren't exact numbers from a study, but we see these things happening. You see loss to follow-up, and on that column, second to bottom column, 0 in treatment X and 6 in treatment Y. And you say, well, you know, that's not very many, that doesn't matter. And then you see consent withdrawal after treatment initiation of 21 in treatment X and 32 in treatment Y. And you go, I just don't know wonder what happened to those patients. What went on with those patients is going on. Those sorts of numbers could significantly impact the believability or the utility of the results at the end of the study. So, little cheer for SWOG here. One of the great things that we do, I think, better than any other group is we have coherent reporting mechanisms across the studies, so we can see what problems happen across studies in an easy way. So this is I think, one of the facilities we have as a group. I guess I must be asking for extra funding. Okay. So here's a study. Here's a SWOG XXXX. Not a real study, let's say. This is a study accrued 3 patients. It's pretty much actively accruing. As you see, you have 2 survival curves again. The blue arm is the experimental. It looks like it's doing better. The brownish arm is standard. Again, survival curves, this is giving you the probability at any one of those time points, 0.51, 1.52. It's giving you the probability of surviving longer than that time. So at any time point, the blue arm is better than the brown arm. And the bottom bar here, I should have should have brought a pointer with me. The bottom bar is giving the numbers of patients censored. By censored meanings, that censored is patients we last saw at that time. So they might be, some of these might be lost to follow-up, but most of them are censored, meaning we haven't followed them long enough. They're just studies ongoing at this point in time. And if we did an analysis of this result, by the way, somebody yell at me if I'm going really too long here, the phase 2 result here, we sometimes, at certain studies, we do analysis at we're doing a larger study, and then we do an interim analysis to see whether we should proceed on. Sometimes they're called phase 2, phase 3s. And so in this study, this data, we did a phase 2 analysis, and we saw the p-value is 0.01. So that was the meaning, oh, this looks like a promising regimen. We should keep on accruing patients. We won't tell anybody about it. We'll just say the study's promising, but we'll carry on accruing patients. Very interesting. So the thing to take away from this curve, the experimental looks better than the standard. Things look promising. Go ahead. This is the situation, same data. So as a statistician, I can play magic sometimes. I can go in and say, oh, what if this data point was different? Let's draw the plots and take a look. So what I did here was I said, oh, I'm going to make 6 of those patients you saw before lost to follow-up. What's going to happen? So now let's look at these two curves. The brown line and the blue line are almost on top of each other. They vary at the end. The conclusion would be, looking at these plots, like, man, this doesn't really look very good. Why are we exposing patients to this experimental regimen that has higher toxicity? We should maybe stop this now. And if you did a phase 2, phase 3, and a look here, the p-value would actually, it's shocking that it's actually 0.1, but it's 0.12. So if your threshold is like it has to be less than 0.1 to carry on, or 0.05, you say, oh, let's just stop it now. Let's not waste additional patient resources on this study, go any farther. If you look in the table in the bottom, it's a little different than before. There's a few more censored here. Here, the censored also means lost to follow-up. You look before, there was 1. Now, so I guess I must have had 7 patients because I see 8. So that's the change happening, these additional censored individuals. So the results is this could make a big difference, right? So in other words, these relatively small number of patients could make a difference in terms of whether, I'm getting excited about stuff when I'm using my hands talking about statistics, so this is always a good sign. Could make a really big difference here. So this differential results, if there's interim analysis, and that's the thing to remember here. When the data are coming in, the data are being looked at ongoing, right? So it's not like at the end of the study we need to get everything right, we need to fix it up at the end. We're making decisions as a group on data that's arriving. So this interim analysis would not show sufficient activity. Well, here's the real story. That's actually the beginning of myeloma studies 0007. There wasn't quite the same analysis plan, but those are those initial patients. We went back and we looked at the data to see what it would have looked like early on, and if there had been that dropout, and assuming that design actually did have an early look, potentially you would have, and we do lots of phase 2, phase 3s that kind of happened after 777, the study happened. We would have missed a positive result. We would have never got to the first slide of this talk because we didn't get that data. That's how important it is. Small study. Big studies are fragile, small studies are more fragile. Even a big study starts out as a small study, and that's what the impact of that is. Here's a randomized phase 2 study, just another example. I won't try to pretend it's not a real study. It is a study, a three-drug combination in GI versus a 2-drug combination. And these were data of the phase 2 analysis of that study. And if you look at it, again, the same plot, these curves look pretty close together. The standard actually looks slightly better than experimental, but you'd just say at the end of this study, there's probably nothing going on with this study. The survival probabilities are about the same. The censoring is about the same. So it supported no further development of that 3-drug combination. So the point here is, now had there been additional loss to follow-up, now you see these curves are farther apart. Sorry for the jittering back and forth here, but I'll show you. Oops. You go back to this first curve, they're right on top of each other, and now the second curve, they've separated. So the point here is, now you notice the experimentals on the bottom. Well, how we manage what's going on in a study, if we actually see the experimental result actually harming patients, of course, as you understand, has big implications in terms of how we communicate what's going on versus just the study saying, oh, it's negative at the end, both drugs were the same. So here's a different story. It's not like we would have missed a positive result. It's just we would have come up with a total action plan that would have been different just because of a differential complete data. So, conclusions, obviously, we need complete patient follow-up. And in the later talks, I'll really get into aspects of both, Keisha will give a talk based on both inappropriate consent withdrawals as well as follow-up, and that'll be great. But here I'm just sort of hitting the overview in terms of the statistical aspects of it. Complete patient follow-up and minimizing the impact of consent withdrawal is critical for study interpretation. Sometimes even studies like the 777, the results are not so massive and robust to differences like that. We show how it could have been affected early on, but sometimes the conclusions are positive, but they're not quite as large a study or other things going on. So it's absolutely critical. As I said a couple of times now, it's not only getting the data at the end that matters. It's getting the data as it's ongoing, because we're making decisions. The Data and Safety Monitoring Committees, these are meeting right now or in a bit, and they're making decisions about whether studies should go on. They're looking at those very tables that I showed you and survival curves that I showed you. There are no good statistical solutions. Smart statisticians are great. There should be a lot of smart statisticians around to make the world a better place, but there's no correction for this. No matter what a statistician tells you, if there is missingness and we don't know why the data is missing, there's no way to correct for that. The only way to avoid a problem is not to have that data missing. And so probably the most important message, as I said, will be to listen to the subsequent talks talking about practical issues in terms of addressing this problem. Anyway, thank you very much. That's all I have for you today.